Welcome to the Deep Dive. Today we're taking you inside what is probably the most critical and complex frontier in biology, mapping the actual physical structure of the brain.
Yeah, we're talking about something called connectomics.
Connectomics. The goal is to understand the complete wiring diagram of neural tissue, right? Right down to the subcellular level.
Exactly. But for decades, scientists have, they've run into a brick wall trying to do this at any kind of scale.
And that wall is really about detail versus destruction, isn't it?
It is. To really understand how the brain computes, you need to see things that are smaller than 100 nanometers.
So we're talking about mitochondria, membranes, and I guess the most important part, the synapses.
The synapses, yes, and communication junctions. For years, the gold standard for that level of detail has been something called volume electron microscopy, or EM.
EM gives you those beautiful high-resolution images, but it has a fundamental physical limitation.
It absolutely does. The electrons you need for the imaging, they get stopped very easily by dense tissue.
So they can't see very deep.
Not at all. Their penetration is limited to just a few hundred nanometers. So if you want a 3D image of, say, a chunk of tissue, you can't just look through it. You have to physically take it apart.
That just sounds like a scientific nightmare. I mean, how do you even handle that kind of disassembly?
Well, they use techniques like serial block-face SM or focused ion beam scanning, EM-FIB-SEM for short.
Okay.
And these methods, they either involve cutting the sample into thousands of sequential ultra-thin slices or using an ion beam to literally mill away one layer of tissue at a time while you image the surface.
So you're destroying the sample as you image it, and then you have to try and computationally restack all those slices perfectly.
Yes, and that introduces so much risk, alignment problems, errors, and it is notoriously slow.
We're talking about, month-long experiments?
Easily, for just one small volume. It works, but it really limits the size of what you can analyze. And that's why the community has been looking for a long time at an alternative X-rays.
Right, because X-rays have this immediate tantalizing promise. They can pass through millimeters, even centimeters of tissue, which suggests a non-destructive large-scale mapping solution.
Yeah.
So why haven't we been using this all along? What's the catch?
The catch is the resolution trade-off. To get the kind of high-res image you need to see a synapse, to get that ultrastructural detail, you need incredibly high-energy, intense X-ray beams.
And intense X-rays are, they're beams of ionizing radiation. They just blast the biological material.
They deform it, they cook it, and ultimately they just disintegrate the tissue.
So we're just trading the mechanical damage of slicing for the radiation damage of high-intensity light. Yeah. Classic scientific catch-22.
It's been the ultimate gatekeeper in 3D biological imaging, and the physics behind this limitation is, it's brutal. There's a law that governs the resolution you can get, and it scales with the fourth power of the radiation dose.
The fourth power. Okay, let's unpack that for a second, because that sounds absolutely devastating.
It is. So let's say you want to double your resolution, maybe from 100 nanometers to 50 nanometers. You'd think you just need twice the dose, right?
Right.
But the fourth power rule means you need 2 to the fourth power, so 16 times the X-ray dose.
16 times? Wow.
And the researchers in the study, they cite an even crazier example. To increase resolution by a factor of 5, you have to increase the dose by a factor of 625.
625. So if your sample falls apart at a low dose, your high-resolution dreams are just over.
Instantly, yes.
Okay, so that explains why mapping a synapse has been locked behind the electron microscope. The challenge isn't the light source, it's the material.
Exactly.
So our deep dive mission today is to unpack the synergistic solutions, this three-pronged attack involving materials, cryogenics, and computation, that finally let researchers break that 625x dose barrier and unlock X-ray tomography for brain mapping.
And let's start with the first, most immediate challenge. Standard sample prep just fails instantly.
What does that standard prep even look like?
So, to maximize contrast in soft tissues, scientists use a heavy metal staining process. It's called R2O.
R2.
And then they take that artisaned tissue and embed it in a traditional hard epoxy resin. It's called EPON. But when the researchers hit the sample with 25 keyed X-rays, the results were disastrous.
Disaster? How? Did it just melt?
Both. Mass loss, and then complete disintegration. The standard EPON resin just could not absorb that energy and hold its shape.
So there was a known limit.
A hard limit, around 6 times 10 to the 8th gray of radiation dose. And they needed to push orders of magnitude beyond that.
Okay, so problem number 1 is structural integrity. The sample needs a suit of armor to withstand the X-ray fire.
A perfect analogy.
Which brings us to the first pillar of this breakthrough, the chemical solution. Where did they even look for something strong enough to survive what is essentially nuclear-grade radiation?
Well, they realized that the usual biological plastics were just not designed for this, so they started screening specialty resins that were developed for the most extreme radiation environments we know.
Which would be what?
The nuclear reactor and aerospace industries.
Wait. They're using space-grade epoxy to image a tiny piece of a mouse brain.
That's pretty much it. It's a fantastic example of cross-disciplinary innovation.
So what was the winning material?
It's a low-viscosity epoxy called TGPAP. And to understand why it's so tough, you really have to look at its molecular structure compared to the standard EPON.
Okay, try and simplify the chemistry for us here.
I'll try. So both EPON and TGPAP are what we'd call trifunctional. They have 3 epoxy groups, which lets them form a really robust cross-link structure when they cure. That's where the strength comes from.
But TGPAP has something extra.
It has a crucial upgrade. It includes a benzene ring and a tertiary amine group. And the hardener they use with it, called DDM, also has multiple benzene rings.
And these benzene rings, these aromatic structures, what do they do functionally?
They dramatically increase the rigidity and the density of the final polymer. I mean, think of it like swapping out cheap plastic scaffolding for reinforced steel rebar.
I see.
These aromatic structures, when they're packed together, they provide just extraordinary mechanical strength and, crucially, phenomenal radiation resilience.
And the result? Yeah. How much better was this new tough resin?
The results were night and day. Where standard EPON failed completely, the TGPAP sample showed almost no mass loss.
None.
Practically none. Just some microscopic localized defects, even when the dose went way beyond 10 to the 9th gray. It held its structure even up to doses over 10 to the 10th gray. That is a massive, massive leap.
That's credible. But for, the average lab trying to use this, how practical is TGPAP? Is borrowing from the nuclear industry going to introduce all these new complexities or crazy costs?
That's a great question. For moving from a proof of concept to a standard tool, the material itself is specialized, yes, but the actual embedding process is basically the same as with traditional epoxy.
So the workflow isn't that different?
Not radically, and the benefit of being able to image huge volumes non-destructively, that probably offsets the material cost when you compare it to, weeks of manual labor for sectioning.
That makes sense. Okay, so we've got the chemical armor with TGP. What was the second part of the defense?
Physical stabilization. They used a system called the OMNY stage. OMNY. And this cooled the samples to cryogenic conditions, below 95 Kelvin.
So why the extreme cold, even with this super tough resin?
Because the cold just drastically reduces all molecular motion, even with the toughest material. That immense X-ray energy still causes movement, thermal expansion, localized changes.
So freezing, it just locks all the atoms in place.
Essentially, yeah. It minimizes any blurring that could be caused by that massive energy dose.
Okay, let's pause and do a quick recap here. To beat this devastating fourth power dose barrier, the researchers engineered a 2-part material solution. First, they swapped out the normal plastic for TGPAP, a nuclear-grade resin with these aromatic rings for extreme radiation resilience. And second, they used the OMNY cryo stage to physically lock the sample down below 95 Kelvin. This let them blast the tissue with the huge X-ray doses needed for high resolution. But even with all that, a kind of ghost-in-the-machine issue remained.
Precisely. Even with the TGPAP and the cryo cooling, when they looked at their first results, the resolution improvement, it just sort of stalled out around 70 nanometers.
Why? I mean, they minimized the damage, but it didn't just disappear.
Correct. The high dose still caused these subtle residual sample deformations. We're talking micro movements, maybe a slight softening of the resin over hours of exposure, or tiny thermal effects.
And these tiny shifts just blurred the final 3D reconstruction.
Exactly. It stopped them from hitting the theoretical resolution limit. So they needed a computational way to fix motion that was happening during the scan.
And here comes the computational gymnastics. This is the third pillar, right? The nonrigid tomographic reconstruction algorithm. How can software fix a physical warp that happened inside a frozen piece of tissue?
Well, a standard 3D reconstruction assumes the sample is perfectly still for the whole scan, the full 0 to 180 degree rotation.
Okay.
The non-rigid approach just throws that assumption out. What they do is they split the full angular range into multiple smaller subranges. So they take several low-resolution subtomograms at different times during the scan.
So instead of one big, long, blurry exposure, they take a bunch of low-res snapshots of the sample as it's deforming, like taking a shaky video instead of one blurry photo.
That's a fantastic analogy. The algorithm then looks at these subtomograms and it estimates the deformation fields, the exact 3D movement or optical flow between those snapshots.
And then it can computationally reverse the warp.
It can. It compensates for the movement caused by the radiation over time, and it fuses all that corrected data into one final crisp image.
That's brilliant. It's like the image stabilization on a modern camera, but for high-dose X-ray physics. So what was the practical payoff of cleaning up this warp?
It was immediate, and it was substantial. For one of their samples, the nonrigid reconstruction improved the resolution from 70 nanometers all the way up to 49.7 nanometers.
Wow.
It successfully recovered those high spatial frequencies that the blurring had hidden. It got them much closer to the theoretical prediction.
And when you combine all three, the TGPAP, the cryo, and this non-rigid reconstruction, what was the ultimate record-breaking resolution they hit?
They reached an impressive 38 nanometers isotropic 3D resolution.
38 nanometers. That is genuinely ultrastructural detail. Now, for those of us who aren't neurobiologists, what can you actually see at 38 nanometers in, say, the hippocampus of a mouse?
Well, this resolution is more than high enough to see major features, axon bundles, dendrites, big organelles like mitochondria and myelin sheaths.
Okay.
But the critical breakthrough for connectomics for that wiring map is that 38 nanometers is good enough to reliably resolve synaptic contacts.
So you can see the actual junctions.
We can now visualize the actual communication points between neurons using this scalable X-ray technique.
All right, another quick recap. The full synergy is this. The nuclear-grade TGPAPs, resin, and the cryo stage let them survive the massive X-ray dose. Then, the non-rigid computational reconstruction cleaned up the leftover deformation, delivering 38 nanometer resolution.
Right. Enough to see the synapses.
It's a monumental feat of engineering.
Yeah. So a question for you then. We can see synapses, but what specific intracellular features, maybe things even smaller than vesicles, do you think would still be a challenge at this 38 nanometer ceiling?
That's the right way to be thinking, what's next. So while we can see the overall synapse structure, things like individual synaptic vesicles.
Little sacs of neurotransmitters.
Exactly. Those are closer to 30 to 50 nanometers in diameter. So while you can see them as a cluster, the membrane of an individual vesicle might still be a little blurry. For that, you'd need to push closer to 20 nanometer.
I see.
But before they could even aim for 20 nanometer, the researchers had to prove they hadn't just created a high-resolution image of damaged brain.
Right. How do you prove the X-rays didn't just scramble the tissue? You use the ultimate ground truth, correlated imaging. After they finished the X-ray scan, the PXCT measurements, they took the exact same specimen and put it through FIB-SEM.
So they took the same piece of brain, blasted it with X-rays, and then physically milled it with the traditional gold standard just to check for damage.
They used EM to validate the X-ray imaging.
Yes. And what did the IFSEM ground truth show?
It confirmed that the X-ray irradiation, even at pretty high doses, did not noticeably impact the overall ultrastructure.
That's huge.
Crucially, subcellular features like membranes, mitochondria, the endoplasmic reticulum, they were all preserved. This validation was absolutely key to proving the imaging was non-destructive.
And how reliable was the synapse identification, comparing the X-ray images to the ground truth from the FIB-SEM?
So they ran quantitative analyses. They had expert annotators blindly identify synapses in the X-ray data. In the lower dose samples, the ones in the traditional EPON resin, they achieved a recall of 65%.
So they correctly found about 2/3 of the true synapses.
Yep. Right. Decent, but, there's room for improvement.
For sure.
And when they switched to the TGPAP samples, which were imaged at a much higher dose, the identification rate got better. The blind analysis showed that over 2/3 of the synapses confirmed by FIB-SEM were reliably recognized in the PXCT images, specifically 70% plus or minus 6%.
And that's the functional proof, isn't it? The ability to withstand a higher dose directly translates into better, more reliable science.
Exactly. The ultimate success here is that TGPAP endurance. The fact that the tissue ultrastructure was preserved even after doses exceeding 10 to the 10th gray, that's the cornerstone. It cements the claim that the radiation damage barrier is, for all intents and purposes, removed.
And removing that barrier is what changes the whole landscape of connectomics.
It does. Because PXCT is non-destructive, the technique is scalable to much larger volumes without that physical stitching headache from EM. The researchers believe this dose resistance is theoretically sufficient for whole mouse brain imaging at sub-20 nanometer resolution.
Sub-20 nanometer in a whole brain volume? I mean, that's a revolutionary concept. That's bringing EM-level detail to the scale of an entire organ.
It is, and they're already exploring variations. They mention things like laminography, which is an imaging geometry that lets them image flat, millimeter-wide slabs of tissue. It could potentially offer even greater efficiency and resolution.
So we've moved from a scientific impossibility to a scalable, non-destructive reality. And with fourth generation high-brilliance X-ray sources becoming more available, this isn't just some bespoke lab technique anymore.
No, it's becoming a powerful new part of the connectomics toolbox, really ready for prime time. It's just an elegant example of how solving one big limitation requires this multidisciplinary approach.
Right.
They had to borrow materials from nuclear engineering, use cutting-edge cryogenics, and deploy these incredibly sophisticated algorithms that correct for movement. All just to let the fundamental physics of X-ray imaging finally work.
It truly removes that radiation damage limit, paving the way for 3D tissue mapping that just wasn't scalable before.
And this is where we want to turn to you, the listener, with a final provocative thought.
Okay.
So, given that this new technique is fundamentally non-destructive, it allows the same intact sample to be used for multiple types of imaging. You can do X-ray, PXCT for the structure, and then maybe something else for the function.
Right.
What are the most exciting potential correlations this opens up for future research? I mean, think about it. If you can map the brain's full physical wiring and then go back and hit the exact same synapses with chemical markers to see their neurotransmitter profile, how does that revolutionize our understanding of diseases like Alzheimer's or Parkinson's, which are defined by both structural and chemical changes?
That correlation between structure and function on the exact same physical specimen, that really is the ultimate frontier. We'll leave you to chew on that one. Thanks for diving deep with us today.